Hey there, this is mini lecture number 1 for Mendelian Genetics, chapter 14. First, I'd like to get into some basic definitions, which you'll need to understand before we get into Punnett squares. Then we'll go into Mendel's research. He was a very good researcher. He studied over 20,000 different pea plants in his lifetime, which takes an incredible amount of dedication and a little obsessive compulsiveness on his part. We'll also talk about recessive and dominant traits, and then get into some ideas that we were ending on in meiosis, which was the law of segregation and the law of independent assortment. And that's talking about how chromosomes separate during meiosis. Then we'll get into Punnett squares, which are fun, and I hope you have fun with some of the problems. Then we'll get into kind of summarizing the chromosomal theory of inheritance, get into human pedigrees, probability, and remember, chapter 15, we talk about human genetics. So genetics talk. A phenotype is a measurable or distinctive trait. Now, sometimes we can actually see the phenotype, like, height or hair color. Other times we have to measure it, like the functioning of a chloride channel. And if the chloride channel doesn't function properly, that may lead to a certain disease, such as cystic fibrosis. So anyway, again, it's a measurable or distinctive trait. A genotype is actually that organism's genetic makeup. Okay, what are the genes that lead to a particular phenotype? Typically, we'll have at least 2 copies of the same type of gene. Okay, so you get one of those copies from your mother and one of those copies from your father, and they would be on homologous chromosomes. We'll talk a little bit more about that later. But I just want to kind of, let you know that oftentimes genes are a little bit more complicated than just, 2 copies. But we'll get, don't overthink that. I just want to mention that oftentimes it's a little bit more complicated than what we're presenting in this chapter. An allele is an alternative form of a gene. Remember, I mentioned you get 1 copy of the gene from your mother, 1 copy from your father. Again, diploid organisms like us and pea plants are also diploid, have 2 alleles, at least 2 alleles for most genes. And each of those alleles is on homologous chromosomes. So again, if we're talking about a gene on chromosome 2, you would get 1 of those alleles from your mother and 1 of those alleles from your father. Okay, an organism that is homozygous for a particular trait is where 2 alleles are identical. Okay, so both alleles in this case, the example, both alleles code for smooth peas. And this could be a dominant or a recessive trait. It just means the alleles are very, very similar. Okay, so we call those homozygous. Alleles that are slightly different, we're going to call those heterozygous. Okay, those alleles are different. So we might have one gene that codes for smooth peas. The other gene codes for wrinkled peas. These are still genes on homologous chromosomes. They're basically coding for the similar traits. It's just one codes for smooth peas. The other codes for wrinkled peas. A gene that is recessive is when the allele is expressed only if it is homozygous. By convention, the recessive allele is symbolized as a lowercase letter. An allele that is dominant is expressed whether it is homozygous or heterozygous. And as we start doing Punnett squares, this will become more obvious. A gene is just a packet of hereditary information. P stands for parental generation, and this is something you definitely want to, remember as we start talking about Punnett squares. P is for parental generation. F1 is first generation of offspring. F2 is second generation of offspring. And with peas, it's usually created by self-pollination of F1. This is obviously something you can't do with humans, but you can do it with pea plants, right? You can self-pollinate pea plants. Character is basically like a phenotype. It's an observable physical feature. A trait is a form of the character, so purple flowers versus white flowers. And a heritable trait is passed from parent to offspring. Again, I prefer to use phenotype in general compared to these, but sometimes you'll see it written in slightly different ways. Okay, again, Gregor Mendel actually wanted to be a teacher, but he was rejected from going to teaching school, so he ended up being a monk. And he studied a lot of mathematics and physics and decided to study genetics. And he did lots of studies on pea plants. He made crosses with 24,000 different plants over 7 years. So we have to get into the reproductive anatomy of a pea plant so you know how peas are self-pollinated. Okay, so the stamen is the pollen-producing organ, carpel is egg-bearing organs. Okay, and I can't believe Gregor Mendel did this with a lot of his plants. So he would take the stamens off the plant, okay, and separate them. And then he would specifically cross certain types of peas. Again, this is parental generation. That's P for parental generation. And F1 is offspring. So he chose to track characteristics or phenotypes that varied with two distinct alternatives, such as purple flowers versus white flowers. He also used varieties that were true breeding. That basically means true breeding varieties had to be homozygous. They couldn't be heterozygous. So these are plants that produced offspring of the same variety when they self-pollinated generation after generation. Again, Mendel didn't know anything about genes at the time. There wasn't a lot known about genes at the time. So in a typical experiment, Mendel cross-pollinated two different types of plants, and this mating or crossing was called hybridization. He always followed traits for parental generation, obviously, F1 and F2 generations. Okay, so with the first experiment, the parental generation, he had true breeding plants. That means those plants were homozygous. He had purple flowers versus white flowers, and he crossed them. And he saw what happened with the F1 generation. And the F1 generation, all the plants had purple flowers. F2, again, so this one, he was going to self-pollinate with itself, okay? And with the second generation, once that F1 was self-pollinated, we had mostly purple plants, right? Purple flowered plants. And then we had some white flowered plants. About a 3-to-1 ratio. So Mendel decided that the purple flower had to be a dominant trait, but the white flower was a recessive trait. He observed the same pattern of inheritance with 6 other pea plant characters. Again, what Mendel called a heritable factor is what we call a gene. So you could see these different types of traits. Okay, so you have seed shape here, seed color, flower position, whether it's axial flowers or terminal flowers, the pod shape, inflated versus pinched, pod colors, green versus yellow, and plant height, tall plants versus short plants. All right, and you can see, so this form of the trait is dominant, round's dominant to wrinkled. These traits are recessive. Yellow is dominant to green. purple is dominant to white, axial is dominant to terminal, inflated is dominant to pinched, green is dominant to yellow, and tall is dominant to short. Again, with F2 ratios, we have these magic, and I kind of add magic in quotes there, magic 3-to-1 ratios of dominant to recessive traits. Now, it was really, really important that Mendel followed these traits for 2 generations. Okay, so you had the parental generation plus F1 and F2. So I guess total of 3 generations, right? But he followed it to the F2 generation. That's really what I'm getting at. Because if he didn't, he wouldn't have seen this 3-to-1 ratio. Okay. So again, alternative versions of genes are called alleles. And alleles are on homologous chromosomes. Also, each gene resides at a specific place on a specific chromosome. And that's really important, especially for crossing over events. So you really need that to happen. Okay, so we're looking at, here is an allele for purple flowers. Here's an allele for white flowers down here. These are homologous chromosomes. These genes are at the same location on homologous chromosomes. Okay. So Mendel's key discoveries. Different alleles account for variations in inherited characteristics. For each phenotype, an organism inherits 2 alleles, okay, 1 from each parent. And again, for most genes, a little bit more complicated than just 2 alleles, but we'll just follow me with Mendelian genetics, okay? Don't overthink it. I'm not going to have you do these crazy, absolutely crazy Punnett squares with, multiple alleles. All right, so if 2 alleles are different, the individual is considered to be heterozygous for this gene. If 2 alleles are identical, then the individual is considered homozygous for this gene. Remember, homo means same, hetero means different. So if 2 alleles are different in offspring, then the dominant allele determines the organism's appearance or phenotype. The recessive allele is quiet and has no noticeable effect or appearance on the phenotype. The law of segregation. When any organism produces gametes, the copies of the alleles separate into 2 different cells. And we know that from meiosis. So this is during meiosis. So an egg or sperm gets 1 copy of the 2 alleles for each gene. And again, we'll reveal the law of independent assortment later, but that it really means that those genes separate into different cells, or the alleles separate into different cells. So let's start working with the Punnett square. So remember, one of the most important things I think, to remember when you're doing Punnett squares is to first make up the possible gametes. Okay, so you're going to only have 1 allele per egg or sperm because it's a haploid cell, right? You do not want 2 copies of the same allele in a gamete. So if somatic cells have, they're going to have 2 alleles, right? So it's GG. The gametes are going to only have, and GG refers to the genes coding for something. The gametes are only going to have 1 copy of each allele, okay? Once you've gotten the possibilities of the alleles in each gamete. Then you can cross sperm and egg genes to see what the F1 generation will look like. And then you can self-pollinate to look at the F2 generation. Okay, so the next mini lecture, we're going to start doing some Punnett squares. All right.